Okay, so the first thing I do is to switch to the desired working directory here. And to list the files in my working directory, I say ls. To list the files in where exactly I kept the raw reads, that is the raw sequencing file, I kept them here, and you can actually see them. My files, another interesting thing about my files is that they are quite small and they are atypical. You're never going to see this kind of, you're never going to see this kind of file on the internet for real scientific projects. They are in the ranks of 392 kilobytes, and usually you should have about 1 gigabyte for humans, at least. I mean, 2 gigabytes sometimes. So, however, the command is the same irrespective of the file size. But the reason why we are using them is because of, for the sake of time and what is feasible within a YouTube video. The next thing to do is to create an output folder where your quality control report for each file will be saved. And to do that, you can just say mkdir QC reports. Sorry, my reports. Yep. So the next thing is to run your FastQC command. And then to do that, you say FastQC, name, for example, raw reads. I'm going to locate the file inside raw reads, SC Barry, which is the first one. I'll be picking the first one for now. Sorry, yeah, R1. And yeah, to specify where the output will be located, you say flag small letter O QC reports. And yeah, it should be completed in 1, 2, 3, 4, 5 seconds. Okay, yeah, let's just wait while it completes and compile our reports. Now the report is compiled. You can try to list the files inside your QC reports, and you have an HTML output and you have a zip output. Now the zip output contains some of the graphicals that was used for the HTML, for assembling the HTML report. Now, because it's an HTML report, you can automatically open it on your Google Chrome browser or Firefox or whatever. And to do that, I have opened mine, and here we go. Yeah. So the first thing you have is your basic statistics. And in your basic statistics, you have basic information such as about the FASTQ file you analyzed, which includes the file name, the file type, the encoding, Sanger or Illumina, the total number of sequences, which is about 5,000. This is like a single gene. And the sequences flagged as poor quality, which is none of them. Sequence length, this is like a perfect data set. As you can see, the report contains one result section for each FastQC module. In addition to the graphical list data provided by each module, a flag of passed, won, or failed. This is passed, this is won, and this is failed. Okay, the red is the fail, the yellow and orange is the warning, and the green is the pass. They are not so reliable with other, specifically they are tuned for good quality, and you should be warned that these flags are threshold used to assign this, the threshold used to assign these flags are based on a very specific set of assumptions that are applicable to a very specific type of sequence data. They are tuned for whole genome datasets, shotgun DNA sequencing, and they are not so reliable for other kind of sequencing data types. Therefore, a module result that has a 1 or flag does not necessarily mean that the sequencing run has failed. Please take note of that. Now we have checked the basic statistics. Let's go to the next module, which is the PAR-based sequence quality. By far, this is probably what most people check out in most FastQC reports. And most people really do not go down to do a lot of the check-ins, but then it's important, right? This is a box and a whisker plot of the average quality score at each position along the read in the file. For starters, you need to know that the sequence in fragment of specific sizes. I mean, you need to know that you sequence in fragments. Now, these fragments have specific sizes, okay? For example, some people sequence in 150 base pairs, some people sequence in 100 base pairs. And looking at this, looks like 100, yeah? So you sequence in fragments. And when you sequence in fragments, this plot is showing you what the average quality score of sequence at each position. Say you sequence in 100, you sequence 100 fragments. Now, 100 base pairs rather, at position base pair number 1 to base pair number 100, this plot represents the average number of average number of quality score, which is reported for the sequences identified at that location. The red line within the yellow box represents the median quality, okay? And then the yellow box is the inner quartile range for 25th to 75th percentile. The upper and lower whisker represents the 10th to 90th percentile score. On the y-axis, which is here, represents the quality score itself, otherwise known as the Phred score, the Phred quality score. There is a link in the descriptions below where you can see a full primer by Illumina themselves. Illumina is one of the giants in genomic sequencing, and they explained both the mathematical and conceptual they give mathematical and conceptual explanation to quality score calculation. So what should you look out for in this plot? Your box plot should be in the green portion, meaning the average quality score is high, right? This green represents safe, red means danger typically, and yellow probably means warning, and you should be careful. And it is still accepted if your whiskers extend into the yellow region, but not so much into the red region, okay? You should take note of that. Basically, from our output, this looks very good, but for instance, you may have some terrible plots, and I can show you what really looks terrible. This is very terrible, right? A lot of the reasonable sequences are falling into the red region, and you can also imagine that sequences after the 100 base pair region, in each fragment have poor quality scores. So that can give you an idea of how to prepare your library as well. Your library can be in the range of 100 base pairs. Some people have been able to do publish very good results with 150 base pairs as well. Okay. Then the next thing is the partile sequence quality score.